shorten telomeres, and it was hypothesized and then has been shown that in fact that might eventually lead to senescence once the cap was lost. So the enzyme telomerase prevents that shortening from happening by adding back DNA to the ends of the chromosomes, and if the processes of shortening and balanced are equal, then you end up with telomeres more or less staying the same size. And so cells can keep dividing because they don't run their telomeres down. Now, there's a side disbenefit to that, a lack of benefit, and that is that human cancers, over 80% of human cancers very much activate that telomerase which keeps those ends, unfortunately, of cancer cells from running down as well. In many normal cells, there's actually an imbalance. So the balance is toward the shortening end. And if that goes far enough, the telomere becomes short enough, and this can be shown in vitro, and many studies in many labs have shown this, to the point where the telomeres, as the cells divide, the telomeres get too short. The telomeres, after a delay, depending on how fast the telomeres run down and how long they were to begin with, then eventually this telomere, which we think of as like the end of a shoelace, the little A goes to the cap at the end of the shoelace, eventually wears down and the shoelace end becomes frayed, the shoelace being, of course, the chromosome. And so that has a direct consequence on cells. A lot of in vitro work has shown this, and unfortunately this process, which often causes senescence, leads to malfunctioning of cells. Now, the uncapping of telomeres has a consequence if the cells lose checkpoints and they become cancerous, and genomic instability can arise. But even if that doesn't happen, the cells are still malfunctional. They don't function properly for their mitochondrial function, for example, and importantly, like rotten apples in a barrel, you can think of those cells inside an organism. They produce substances, pro-inflammatory factors, pro-tumorigenic factors, that can affect other cells. So they're not only a danger to themselves, they're a danger to other cells. So, the problem in humans, and much research has shown this, is that gradually over our, say, 80 plus or so years, we hope, of life expectancy, as one might observe at the best situations in developed countries, telomeres gradually shorten from about 10,000 bases down to about roughly half of their length at the point where most people will die at some elderly age. So we have this issue of this is really going on in humans, there's evidence this happens in people, and the question is this really of consequence? Is it, metaphorically speaking, to change the metaphor, as though the candle is burning down in some way? Counteracting that process is that there is some graded amount of telomerase in cells. There is a certain amount of cells that have telomerase in them, stem cells, for example. Certain number of, say, germ cell lineages, of course, those cells have it, and a certain number of other cells have a certain amount of telomerase, but nevertheless, there's evidence that this process does get out of balance over the very decades-long lifespans of humans. And so there was no real way to predict from animal models or in vitro studies how this was going to play out in humans until one looked at humans. Unfortunately, a great deal of work from a great many different research groups around the world has now really started to collapse this into, I think, a situation where we can get the picture. So I'm going to say what this kind of research has told us about, and I won't be acknowledging the very large number of research groups who have contributed and made major discoveries in this. I'll just make the summary points here. Because what we want to know is if there is reduced telomere maintenance in humans, as we can see gradually happening over lifetimes, what's the impact of it? And much research has given us a pretty good picture, and that is particularly genetic influences have been informative, but they may not be the major influences, but they've given us proof of causality that, in fact, the things that impact on telomeres and make telomeres get shorter do indeed change the risks for aging-related diseases. And as I'll show you some data, actual data, mortality. So that's become very clear from large cohorts about mortality risks. And so again, all these often comorbid conditions of aging are influenced by telomere shortening. A lot of studies indicate all of this. And there's causality in there, but there's also other factors, of course. In no way do I want to suggest that this is happening in most human beings in isolation from other processes and other inputs, both genetic and the multiplicity of non-genetic inputs, many of which are the major topic, as we heard, of this conference, or this Congress. And it can be a two-way procedure as well. And so there's good evidence that duration and severity of depression, for example, feed back on telomere attrition, exacerbating it. So, just give you an example of the mortality results, and what you see in a real sample of adults when you measure telomere length, which we did. We made a robot to measure 100,000 people's worth of telomere length in a very large study in California, the Kaiser Permanente study, the JERA cohort led by Kathy Schaefer and Neil Risch. And this is a brief description of some of the points about it. And the point is that it had 100,000 people in which we could get very good low coefficient of variation measures of telomere length, actually using saliva samples, a very mixed cell type, and there are a lot of data, including biospecimens, a lot of clinical data, a lot of survey data about these individuals and the GWAS data. So I'll just share with you a little bit of the telomere data. This is what we saw when you look at adults ranging from about 20 up to about 90 in this graph, and what you see is the decline. It looks a little bumpy on the left for technical reasons. That's not to do with the accuracy, but actually young men don't like to give samples, and so we undersampled young men. But in fact, what you see is a rather interesting split, which hadn't been seen before because studies hadn't been powered this high, between males and females, which occurred postmenopausally. So interesting biology. But also interestingly, after a kind of nadir, at the age of maximum mortality between 70 and 80 in this cohort, roughly around 75, then there was actually an increase in mean telomere length in individuals, suggesting some selective value, since most people living beyond that are actually living beyond the age of the highest mortality in the mid-70s. Now, another piece of data one could get was to look what happened subsequently, because the samples were picked up, analyzed from people that had given their sample in 2009. One could look at all the death records, mortality, and say what had happened 3 years later, and who had died in the intervening 3 years, and then compare that to what the telomere length had been at the beginning. And so the answer was that there was a very clear relationship, which was that the people in the bottom quartile of telomere length had a higher risk, and you can see it on the graph, of mortality in that 3-year period. Now, we know a lot of things influence telomere length, and I'll talk about those a bit more. And so many of those actually are themselves risk factors, and I've listed them here: age, gender, race, ethnicity, education, cigarette pack years, physical activity, alcohol per week, BMI. And so we could adjust for all of those. And what was very interesting was that it didn't shift this relationship, this risk relationship at all. So in other words, telomere length was an independent predictor of increased mortality, and quite a substantive effect size, as you can see here. And then, just to show that we're not crazy Californians, beautiful study out of the Copenhagen study, and I've just graphically shown their results, which is again after all the adjustments, the multi-variant adjustments have been made, looking now across the deciles of telomere length. So the shortest telomeres are the ones on the right, well, on your right, and what you see is that again there's a clear difference between the longest and the shortest telomeres in terms of mortality risk. In their case, it was an average of a mean of a 7-year follow-up. So telomeres are interesting. They predict mortality. There's good genetic data, which I haven't had time to go into, but in which mutations that recur in humans that take telomerase down a notch, down to half its normal level, or which interfere with those telomere protective proteins, clear monogenetic diseases, showing clear causality of lack of telomere maintenance in causing a set of diseases that include, interestingly, cardiovascular disease, as well as, interestingly now it's been clear, diabetes and aplastic anemia, pulmonary fibrosis being prominent among them, and certain kinds of cancers as well. As I said, the genomic instability that's predicted from telomeres failing to cap telomeres would be expected to have a readout in terms of cancer risk, and indeed such things are found for certain subsets of cancers in the individuals with the monogenetic diseases. So then these other correlations that I've shown you are quite suggestive that causality can exist. And there are some population studies, which I won't have time to discuss, but which when one looks at telomere gene variants, common variants in the population at large, one does find that this can be related to risk for example, coronary artery disease. A beautiful study by Codd et al. In 2013 made that clear. So there is some causality one can infer about telomere maintenance in the general population as well. And interestingly, coronary artery disease is a good example where one sees an influence which is genetic in origin, and therefore it's very hard to escape the idea that it's playing some causal role in all of this. Not by itself, of course, there's probably a lot of interaction going on. So what might influence telomere length? Well, it's long been known that aging is not unrelated to stress. And a wonderful story from 2,500 years ago of Wu Shizhu, who was a Chinese very high figure in one of the provinces of China, and he was having to cross a dangerous border between two different provinces of China ruled by different powerful forces. So he had to cross this extremely dangerous border, and his hair overnight went white with worry. And the good news was that he was unrecognizable, and so he got across the border safely. But the example was that here's an example of rather extreme stress causing, you know, this idea of white hair overnight. So the idea of stress and aging related phenomena are certainly not a new idea. So what about stress? Well, as we've already heard alluded to, stress is well understood to play a role in impact, and cardiovascular disease is a very good example of that, of which there's excellent studies. And so what we've been finding is that, in fact, again, from a number of studies from our lab, and again from a number of different research groups, is that you do find a relationship where what one sees is an input, which of course is the chronic stressor from whatever situation, but when certainly acting on the brain, there'll be a lot of physiological interaction, processing, if you will, of the signal, a lot of physiological changes resulting from what goes on when someone is chronically stressed, and a lot of this is understood. But what we find is that there is indeed a relationship, and it looks very causal at this point, on telomere shortness. And as I said, telomere shortness, telomere lack of maintenance can have an impact on disease, as we've discussed, and it's shown genetically in the more extreme cases and in more limited cases in more common situations. Now, of course, that won't be acting alone, but it's certainly a pathway, and there's good reason to think this is part of the pathway into how chronic stress does impact on common diseases. And I just want to list the kinds of studies. This is by no means a complete or even up-to-date today list, but it gives you an idea of the breadth of the kinds of chronic stress or situations that have been seen. First of all, looking at shortened telomeres in adults, and they range from things like perceived stress, particularly strong in caregiver situations when there are objective stressors, and then extending through major depression, as I mentioned, dose dependency, severity dependency, even things like former domestic abuse and stress load, social stress load, and cumulative exposure to childhood traumas, and even less education really goes across multiple cohorts with shorter telomeres. So all of these are starting to suggest where larger policy decisions need to be informed by information of this kind, because it extends to children as well. And now there are studies which are really showing, if you now look at that critical stage of development in children, where telomere shortening takes place, which may be quite decisive for adult telomere maintenance. And so now a number of studies have related it to, for example, severe emotional neglect, exposure of the children to violence, and so forth. And so, and even environmental, socioeconomic issues like neighborhood criteria. And I just listed again a few of the studies here. And it goes even earlier. It's beginning to look as if one can see influences of maternal situations, both stress and nutritional, on the telomeres of her newborn, and in one small but provocative study, even the telomeres of children who've grown up to be 20 years old. And so this related to a very severe historical stressor during the pregnancy of the mother. So I think all of these start to have implications in terms of thinking more broadly about policy issues, health policy issues, socioeconomic issues, and so on. Now, I mentioned that shorter leukocyte telomeres are an independent factor, and I've just put up Willite et al.'s study, which first of all beautifully showed this. This is a European cohort, and what it's showing is telomere length tertiles, and the x-axis is indicating the telomere length tertiles. The shortest telomeres are at the top, and the longest telomeres at the bottom, and looking at different categories of cardiovascular disease over the succeeding 10 years after the telomere length was measured. That's right at the beginning of the x-axis. And so what you see in a graded way is increased risks of incidence of these various forms of cardiovascular disease, and also of certain mortality categories. So it's anticipating that. And so okay, so that was one kind of prediction, but can we do better? Now, what I want to end with is just a very interesting thing that's come from the world of cancer, where the kinds of risk factors I've been telling you about and looking at in terms of telomere length being measured as a risk factor are sort of very statistical. They're not that clinically actionable. But a really exciting action, I think, is coming out of cancer, and I'm going to just give you one example, which is a study from MD Anderson in Houston, Texas, and what was done was to look at a cohort of bladder cancer patients. And so what they did was that at the time of diagnosis, they'd come in, they're clinically diagnosed, it's a whole variety of people, 440 people, and at the point of diagnosis, they were categorized into either having depression on a standard depression scale, short telomeres, that median split, lower half, upper half of telomere length, in their leukocytes in blood draw, not the cancers, just blood draw telomeres, as is very commonly measured in all of these cohorts I've mentioned, or neither of those, or both of those. And so I'm going to, for the aficionados, that's the official Kaplan-Meier survival curves, and the point is that they've adjusted this, what they're showing here for all these sort of possible confounds, including the degree of cancer, the stage, the type, and so forth, and treatment modalities. But what I want to show you is a much more visual version of that. So here we are at diagnosis, everybody's alive, 440 patients, so a whole variety of different people. And now what is done is to say, if you, and this is just showing in a more graphic way that Kaplan-Meier survival curve I just showed you. If you said who after 2.5 years had died, so that's the black ovals, and so that would be the people who only had depression or only the short telomeres who had neither, there was not much statistical difference between those three groups. But at the same time point, if you were in the group of people who had both the shorter telomeres and the leukocytes and you had depression, then this fraction had died. So there's a big difference. Now we go to 5 years, which in cancer treatment is a real milestone. So of course, after 5 years, many more people have died of the single depression only, or only shorter telomeres than leukocytes, or neither. But of the people who had both of those at the time of diagnosis, this was who died. So you see the point, that what we're seeing is the two rather weakish things, depression, which certainly has a statistical relationship to bladder cancer mortality, and telomere shortness, which again had a much weaker, in fact, relationship to bladder cancer patient mortality. But when the two were combined, those who had both, there was a significant increase in, as you saw, the rates of mortality attributable to bladder cancer in this case. Now, of course, you all know here that depression is also related to coronary heart disease. And just to sort of expand very slightly on that statement, it predicts development of coronary heart disease, there's increased mortality associated, and so forth. So there's clear associations of depression as well. And so it'll be very interesting, I think, to ask that same kind of question in the setting of, say, cardiovascular disease in various forms. What if you now combine these factors and to ask, will there be some sort of interaction? So I think this is going to be an important thing, maybe one can even add more. So this is going to be an important direction, and I think might justify the combination. So, thank you so much. So, a pause, right? So this is all the bad news. What about good news? So, again, I'm going to summarize the mini studies from mini groups, and I apologize that I haven't given all the due credit to the many groups who've done this. But we can ask, what sorts of things can we see? And these are based on observational studies with all the limitations that implies, but they're still pretty consistent and suggestive about things that pull telomere length maintenance down in the bad direction. And chronic stress, I've listed a set of various subcategories, and on the good side, the kinds of characteristics that have been seen in observational studies. So it looks as though there's a malleability to telomere length maintenance, and the big challenge is going to be seeing what actual proper randomized placebo-controlled prospective trial can tell us. That kind of study can tell us about what is the best way to intervene in ways that are most effective. So the observational studies have certainly pointed the way to go, and certainly give one some very useful information, which, by the way, doesn't look an awful lot different from what your doctor, your cardiologist, your family doctor, your general practitioner, or your mother told you, which is, you know, eat right, sleep well, you know, keep a good attitude, reduce your stress, exercise, primary exercise. And of course, as we go into much more genomic information, how will these things interact with genomic information? Big open questions. So where we are is that we do find that there's evidence that telomere attrition is acted on by a variety of factors, of which genetic gives us proof, but the quantitatively larger effects, it seems, are actually the non-genetic ones. We can see impacts on telomere maintenance in our cohort studies, and we meaning a very broad number of people. And again, this is through a lot of interactions with other factors. It's a two-way street. This is not straightforward. There's a caveat, which is that cancers have a complex relationship to all of this, because genetically pushing telomere maintenance up, which is good in all other respects, is actually now clearly a risk factor for increasing certain of the minor but serious cancers like melanomas and gliomas, and even a certain subset of lung cancers. So there's a nuanced situation, although overall it looks as if, from all the data, maintenance of telomeres through at least the kinds of physiological and other sorts of interventions or things that I referred to in the previous slide, all of those seem to have positive outcomes in terms of mortality and everything that can be measured as in terms of observed telomere length. But we have to be well aware that pushing up telomerase in certain ways is probably playing with fire, and we have to be super careful about interventions in ways that might push things towards cancer risks. So I just want to finish with acknowledging the fantastic collaborators that I've had, of whom I've listed a few major ones, and I have many more people to thank. And my research group, wonderful people, and particularly Lynn takes the pride of place as being the person who has driven many of the studies relating to the collaborations with clinical studies that I've been mentioning. And so I want to thank you for your attention. Thank you.